This video will cover action potentials and conduction in neurons. Neurons carry information. Axons transmit information from the cell body to axon terminals, which communicate with target cells at synapses. The difference in ion concentration across the membrane creates the resting membrane potential, or RMP. Changes in membrane potential produce the electrical signals that transmit information. Depolarization is a decrease in potential difference. Hyperpolarization is an increase. Repolarization returns to the RMP. Overshoot is when the inside of the cell becomes positive. Neuronal signaling involves gated channels. Graded potentials travel short distances and are activated by mechanically or chemically gated channels. Action potentials travel long distances and use voltage-gated channels. Graded potentials are depolarizations or hyperpolarizations, proportional to the strength of the triggering event. They lose strength as they move through the cell due to charge leakage. A graded potential depolarization is an excitatory postsynaptic potential, or EPSP. A hyperpolarization is an inhibitory postsynaptic potential, or IPSP. They occur in the cell body and dendrites. Local current flow is the wave of potential moving through the cell. Graded potentials travel to the trigger zone. If they depolarize the membrane above the threshold voltage of about -55 millivolts, an action potential is triggered and travels down the axon. This slide is blank. A neuron receives many inputs. Spatial summation is the initiation of an action potential from several simultaneous subthreshold graded potentials from different locations. Temporal summation occurs when summation results from graded potentials overlapping in time. Summation demonstrates postsynaptic integration, a key property of neurons. Action potentials are all or none events when graded potentials exceed the threshold. They maintain the same size over long distances and are identical. They occur through alteration of Na+ and K+ ion permeability via voltage-gated channels. This slide compares graded potential and action potential. This slide illustrates the time course of the action potential. Sodium channels have two gates, as shown in this figure. The inactivation gate's movement is coupled to the activation gate, but is slower. The Na+/K+ ATPase has no direct role in the action potential. This slide is blank. During the absolute refractory period, a second action potential cannot occur until the first is over, as Na+ gates have not reset. The relative refractory period allows a large suprathreshold graded potential to start a second action potential. Absolute refractory periods prevent backpropagation of action potentials into the cell body. Refractory periods limit the signal transmission rate to about 100 impulses per second. Action potential movement along the axon is called conduction. A wave of action potentials travels down the axon, with each section in a different phase of the action potential. A graded potential triggers an action potential, opening voltage-gated sodium channels. The sodium spreads in all directions, opening sodium channels and initiating an action potential in the adjacent region along the axon, but not in the cell body. Potassium channels have opened in the initial segment. Sodium ions cannot trigger an action potential there, as it is absolutely refractory, but they initiate action potentials further down the axon. Action potential conduction speed depends on membrane resistance to current leak and axon diameter. Larger diameter axons offer lower resistance, speeding up conduction. The myelin sheath insulates axons, preventing current leak. Nodes of Ranvier are gaps in the myelin sheath with a high concentration of sodium channels. Saltatory conduction is the apparent leapfrogging of action potentials from node to node. When depolarization reaches a node, sodium enters, reinforcing the depolarization and maintaining action potential amplitude. In demyelinating diseases like multiple sclerosis, the loss of myelin slows down action potential conduction, leading to symptoms like muscle weakness and vision loss. These are the references used for this video. The frequency of action potential firing is proportional to the size of the graded potential. The amount of neurotransmitter released is proportional to the frequency of action potentials.